Hello and welcome. I am Therese, the CTO of Biocrates. Today I would like to show you how common population disease develops via alteration of the metabolism. Here I will discuss two hot topics: the role of microbiome for gut health and the need of drug development for COVID-19. Secondly, I will set the focus on the importance of broad pathway to deep pathway analysis on the example of the underestimated relevance of the tryptophan and acylcarnitine metabolism. Over the past years, we could develop an improved understanding about the complexity of metabolic diseases, the role of pathophysiological processes in the disease development over 10 to 20 years, and the underlying risk factors as the exposome, genetics, and individual metabolome and lifestyle factors. The metabolic risk factors as metabolic syndrome, low-grade inflammation, are quite dominant as drivers in the metabolic reprogramming and disease development, for instance for cardiovascular and autoimmune diseases. The three major clinical needs for these metabolic diseases are: first, to find predictors and early detectors to avoid or delay the disease development; second, to develop improved diagnostic tests; third, to push precision medicine research for patient stratification, therapeutic intervention, and finally, in prognosis and outcome. I am convinced you agree Metabolomics is a technology of choice to better understand metabolic disorders. It is important to get new knowledge to better understand the metabolic commonalities and specific differences of the disease development. The readout of the individual metabolic phenotype explains the function and deregulations of processes. As an example, insulin resistance can be detected by amino acid and acylcarnitine metabolism deregulation, or gut dysbiosis by the analysis of the metabolic alteration of bile acids. Interesting to note, these pathophysiological processes are linked to cellular reprogramming resulting after years in disease manifestation. The first spotlight is metabolomics and the relevance of the microbiome. The gut-liver-brain axis connects organ as main communication channel. Why do we carry 1.5 kilograms of gut bacteria? What is the function? How the food intake affects the gut microbiome and gut health. The gut microbiome plays an important role in many diseases with high clinical need for prediction, development, diagnosis, prognosis. Therefore, we need suitable technologies for gut microbiome analysis to better understand the link of the gut to the other organs, and finally the contribution of the microbiome to the human health. There is a technology named as 16S RNA sequencing, which analyzes the bacteria presence and composition. But to get more insights into the effects of the microbiome dysbiosis, we need to analyze the functional readout, where metabolomics is a technology of choice to better understand the high degree of crosstalks and communication channels. A central question: which is a sample matrix of choice in microbiome research? I say feces and blood. Both have an utmost clinical value, and blood should not be underestimated for microbiome health analysis, because the circulating metabolites in blood can tell you a lot about the nutrition-host microbiome interactions. Is there a need for new clinical tests for gut health analysis, or can we translate established clinical tests? Yes, there is a need. This study demonstrated that established clinical test parameters, 77 were tested against metabolomics, are not able to accurately predict the gut alpha diversity. The results show that with only 11 plasma metabolites, a much better predictive value was obtained compared to the clinical lab test. In addition, potentially 40 metabolites with a metabolic link to cardiovascular diseases, diabetes, and kidney function can explain 45% of the microbiome variance. These are promising results and show that the ability of the blood metabolome to predict gut microbiome diversity could pave the way to the development of clinical tests for monitoring gut microbial health. This figure shows you the knowledge today on how gut microbiota and derived microbial compounds may link to metabolism of the healthy host or to the pathogenesis of common metabolic diseases. An example: dietary phosphatidylcholines from meat or high-fat dairy products is a primary source of choline, Choline degradation to TMA occurs by bacterial enzymes. TMAO is formed by N-oxidation by supportive FMO3 liver enzymes. Increased TMAO blood levels reflect renal medullary damage and is independently correlated with cardiovascular diseases like atherosclerosis. Atherosclerosis is one of the leading causes for morbidity and mortality in the Western world. People have prior clinical silent metabolic dysfunctions for many years, including elevated circulating concentrations of glucose, insulin, lipids, in addition to insulin resistance and low-grade inflammation. I see that the better understanding on how microbial messengers regulate the host metabolism is also pushing the identification of new drug targets and mechanisms for drug toxicity, metabolism, and efficacy. My second spotlight today are infectious disease, specific on COVID-19. Infectious diseases have a very strong link to metabolic phenotype and especially pre-existing metabolic risk factors present in the human being drives the infection severity, as we already know for COVID-19 today. On the right-hand side, you can see the disease progression and different disease severity stages of COVID-19. There's a high medical need to correctly detect ad hoc the infection, to stratify patients, predict and monitor the therapeutic responses and the progression. Luckily, we can learn a lot from known infections like community-acquired pneumonia, sepsis, and MERS, also about the metabolic drivers and commonalities for COVID-19. We know these metabolites and the pathophysiological roles, for instance, in immune modulation and inflammation. Therefore, I see metabolomics with high potential for market development for all the clinical needs discussed here. No effective antiviral COVID-19 agents for treatment are currently available. Therefore, we have not only high medical need for correct diagnostic tests and vaccine development. Improved treatment options are also very important. Here we have to understand the complexity of diseases more in detail. We know COVID-19 as a new pathogen need antiviral-based anti-inflammatory and immunomodulation strategies. Have a look on the metabolic risk factors for COVID-19, like type 2 diabetes, obesity, hypertension, immune dysfunction, and chronic low-grade inflammation. Together with the virus infection, these risk factors are becoming dominant in severe disease stages. Respiratory failure can result in hypoxia and subsequent damage other physiological systems. Cardiovascular damage can limit transport of oxygenated blood to other organs. And finally, kidney injury supports the accumulation of toxic metabolites in blood, which are transported to other organs as well. The following cycle shows you some of the major metabolic drivers in COVID-19 infection and their role and effects, for instance, lipid and glucose metabolism in viral replication or antiviral response. Surfactant lipids are mandatory for respiratory function. Mitochondria and therefore acylcarnitines are important for pro-inflammatory cytokine production. And fatty acid metabolism is highly relevant for cell repair. Targeting the lipid metabolism in infected host cells could be a potential new antiviral treatment strategy here amongst established therapies as anti-IL-6 therapy to block the cytokine storm in severe COVID-19 patients. The Biocrates technology offers you here the right tool to improve the metabolic understanding and be supportive in treatment development. The Biocrates flagship technology, the Quant 500 kit, is a standardized and ready-to-use product delivering an excellent tool for targeted metabolomics, with broadest coverage of metabolites and lipids in the metabolic network and pathways. In addition, we have supportive kits and assay solutions available for you, addressing a deeper look into specific pathways. The Quant 500 kit allows a quantitative analysis of up to 630 metabolites and lipids with a coverage of all key metabolic pathways and allows not only the systematic analysis of nutrition microbiome host interactions. With only 10 microliter sample volume, you can analyze from cell supernatants to animal to human blood and tissue samples. Globally, first international ring trials in metabolomics have proven the robustness, analytical accuracy, and availability to pool the data. The new software tool, the MetaboINDICATOR, at the readout of additionally 230 metabolic sums and ratios. The MetaboINDICATOR supports you in better understanding and interpretation of your data. A combined consideration of metabolites and lipids brings these benefits: increased confidence by higher statistical power, advanced pathway interpretation to read out specific enzyme activities, and ratios as applied in newborn screening to normalize inter-individual differences. Quantitative analysis is the key to success of biomarker signatures, to develop them into future clinical application, and to develop reference ranges and biomarker cutoffs. The Fisher ratio, based on branched-chain amino acids and aromatic amino acid, is an example for MetaboINDICATOR in blood for liver failure severity. The presented Quant 500 kit is the approach of choice to develop and to validate your hypothesis. We see the need in the research community to have in addition solutions for deeper look into specific pathways, with broader coverage and improved sensitivity, to verify your hypothesis and get more insight into the pathway cascades. Therefore, we have developed the here listed relevant pathway-specific kits and assays for you. I would like to highlight two new Biocrates products addressing the tryptophan and acylcarnitine metabolism in more detail. Tryptophan is an essential amino acid and need to source by food intake. The tryptophan metabolism is very special. 3 pathways are linked to the tryptophan with high impact on immune modulation, inflammation, gut motility, gut-brain signaling, gut-brain integrity, the kynurenine, serotonin, and indole pathway. The tryptophan metabolism is also very attractive as therapeutic drug target, for instance, for cancer therapy over IDO. Cancer seems to hijack this mechanism by upregulating the tryptophan kynurenine pathway through the overexpression of IDO. Many companies have therefore developed IDO inhibitors. The newly developed quantitative LC-MS-MS-based runs with 100 microliter sample volume, 17 metabolites as described in the pathway were quantified for advanced pathway analysis. The detailed analysis of the kynurenine pathways shows high relevance, for instance, for Parkinson's disease development. Scientists have shown that the pathway is shifted to the neurotoxic side and the neuroprotective side is going down. This offers the potential to develop signatures for early detection of Parkinson's disease. The acylcarnitine metabolism is a mandatory key pathway to make life possible in general. Acylcarnitines are strongly linked to mitochondrial health and energy homeostasis. They are responsible to transport fatty acids into mitochondria for fatty acid oxidation as a major source of energy for cell activities and further through metabolism of glucose and amino acids. Mitochondrial dysfunction, characterized by a loss of efficiency in the energy production or imbalance in energy homeostasis, is a major factor for developing chronic diseases like cardiometabolic disorders or cancer. Since the level of plasma acylcarnitines reflects the composition of the acylcarnitine pool within a cell, plasma is an ideal matrix to read out the mitochondrial function. In 20 microliter sample volume, up to 44 short, medium, and long-chain acylcarnitines can be analyzed with excellent selectivity of isomers and isobars. For instance, iso-C4 and C4 can be separated, which is very important in newborn screening to distinguish between different diseases. In conclusion, there are many open clinical questions for metabolic diseases, from prediction to diagnosis and therapeutic treatments. We know today metabolic diseases are very complex, addressing variances of pathways and physiological processes. Therefore, the future lies in metabolic signatures, where Biocrates deliver the right technology for you. Thank you very much for your attention.